So the first part to think about is this way of thinking about the plant life cycle that we call the alternation of generations. We have two separate phases, the haploid gametophyte phase and then the diploid sporophyte phase. What's important to remember about this is that these are both multicellular parts of the life cycle. They don't necessarily look like one another, but they are this part of the plant life cycle. And you can see what's happening here is that the gametophyte, so this multicellular organism or part of the life cycle, it undergoes mitosis to produce the egg and the sperm. So there's a first big difference there between plants and animals, is that gametes are produced by mitosis and not through meiosis. These gametes will come together, so through fertilization, that makes a diploid zygote. It goes through mitosis, makes an embryo, eventually becomes a multi-celled diploid sporophyte. This sporophyte will have a structure called a sporangium, plural sporangia. Inside of this structure, there will be a type of cell called a spore mother cell. It is a diploid cell, and that's what undergoes meiosis. So the spore mother cell undergoes meiosis to make haploid spores, and these spores divide through mitosis to make a multicellular haploid gametophyte. That gametophyte, we're back at the beginning of this cycle, it can use mitosis to make the sperm, the eggs, the gametes that fertilize to move on. And so when we are looking at the phylogeny of plants, as you can see here, one of the big changes that is occurring is we are starting off with the earlier diverging, some of these older lineages that diverge from early on in plant evolution, the bryophytes. These have a dominant gametophyte. So that means that gametophyte is photosynthetic, it can live on its own, and the sporophyte gets all of its nutrition, water, and everything from that gametophyte. When we get to this next group, seedless vascular plants, both the gametophyte and sporophyte can live independently, but for a period of time, the sporophyte gets its nutrition from the photosynthetic gametophyte. By the time we get to the seed plants, which includes the gymnosperms and the angiosperms, what's happening now is we have a dominant sporophyte, and the gametophyte is not photosynthetic anymore, and it's nutritionally dependent on the sporophyte for it to survive. So we've switched things as we start from the older bryophyte lineages up to these more recently evolving or diverging angiosperm and gymnosperm seed plant lineages. So let's also think about this in terms of what you need to be thinking about to organize these different groups. Looking at it this way, so the bryophytes are composed of the liverworts and mosses. There's another group called the hornworts. Don't worry about them. Within the seedless vascular plants, the lycophytes and the pteridophytes, so the ferns and the fern allies. We'll see some pictures of those in a little bit. The gymnosperms, several groups, the cycads, the ginkgos, the conifers, and then another group, the neophytes, that you don't need to worry about. And then our angiosperms, three groups, the basal angiosperms, monocots, and the eudicots. And I'll show you some pictures of all of those as we go through this. But just to start off with, here is an image that shows you what bryophytes look like with the photosynthetic gametophyte and the sporophyte structure attached to it. Here's our fern. So the gametophyte is tiny. You can see this relative size difference, the little bitty thing, as compared to the sporophyte. By the time we get to the seed plants, so the gymnosperms and the angiosperms, these large sporophytes, and then very, very small gametophytes, as you can see here and over here. And we'll look at some micrographs of those in just a bit. So let's start with our non-vascular plants, what are collectively called the bryophytes. Here is the alternation of generations, and you can see the diploid gametophyte stage here, a separate male and female gametophyte. So the female, she makes eggs. They're contained in this structure called an archegonium. The males make sperm in the antheridia, and these sperm are released, and they actually swim through the environment. So they have got to swim through a thin layer of water that's on these plants, all the way from the male gametophyte, swimming all around, eventually coming to the female gametophyte, that will swim down this canal and fertilize the egg to make the zygote. So that zygote, it's our early sporophyte stage. You can see how it develops and ultimately grows out of the gametophyte. And this structure up here is the sporangium. That's where the spores are going to be produced. They're eventually released through mitosis. They germinate, grow to give us our male and female gametophytes. Here's a picture of a moss, a typical moss that has the gametophyte here and the sporophyte growing out of it. Here's some other just pictures of other types of bryophytes and some of their general characteristics, which you should think about as being shared by all of these different types of bryophytes. Here's some micrographs just showing you what this all looks like. Here is a female, and so we've got the archegonia up here. Here's a close-up. You can see the egg cells right in there. And here's a male, so these are all the sperm being produced in the antheridia. This plant is a liverwort, another common type of bryophyte. And in this example, you can see the gamete-producing structure of the females growing up out of here. And what's going to happen is that the eggs that are contained on this structure will be fertilized, and the sporophyte will be produced sort of dangling down underneath. Here's a micrograph showing that, and it's full of all of these spores that will be released and produce another liverwort like this. Our next group, the seedless vascular plants. So the big evolutionary innovation here is vascular tissue. And once you get vascular tissue, you can make stems, you can make roots, you can make leaves. So all of those great things about plants are now possible because you've got vascular tissue, which can obviously transport things around the body. But remember, xylem is rigid. It has that lignification, which makes it structurally strong. So this is giving you not only a vascular system, but a structural support system. So anyhow, here are some of the defining traits of different types of seedless vascular plants. Over here, we've got some horsetails, a common plant in wetlands. An actual fern. Here's this is called a fiddlehead. It's the sort of structure they make as they unroll. Here we've got club mosses, and this diagram, or actually this picture up here, shows you the stem and the tiny leaves and the spore-producing structures up there. And then finally another fern. This is a common fern called Christmas fern. You can find all over the forests in Oklahoma. The life cycle is shown here. Pretty straightforward as compared to what we've seen before. We have a gametophyte. Here we can have male and female structures on the same gametophyte, but the basic idea is that the sperm are released, they're motile, they swim towards the eggs that they fertilize to make the next zygote. And so here's that sporophyte embryo growing up out of the gametophyte. This picture shows what that looks like in real life. And then this will eventually become a fern, which has its sporangia underneath. And you can see the structure here that releases the spores to start this cycle again. Here's just a close-up showing you what that female gametophyte of a fern looks like. Well, I guess it's a hermaphroditic gametophyte because it has both the archegonium and the antheridia. And here's a sporophyte growing up out of that. This is the spore-producing structure that you'll often find on the underside of the leaves in ferns. This is a structure called asaurus. And so the asaurus, as you can see here, it's got like a little covering. This micrograph shows that. But underneath, this is where we have the sporangia. And so inside these sporangia, these structures, those are the spores. And so the saurus, plural sauri, but that's where the spores are produced on your fern leaves. These are vascular plants. So just to show you a couple of cross sections through the roots and stems of different club mosses and ferns and things, you can see the xylem and the phloem. Again, xylem and phloem here inside these vascular cylinders inside of the stem. So clear evidence that these are vascular plants. Here's a close-up of one of those club mosses, what their sporangia look like. So lots and lots of spores that are produced inside of there. And that brings us to the seed plants. So the first big group of seed plants we think about are the gymnosperms. These are things like cycads, ginkgos, conifers, the cone-producing trees, and another group that's probably not as familiar to you called the gametophytes. And I'll show you some pictures of those in a little bit. But the big thing here is that we have a dominant sporophyte generation and a very, very small gametophyte. So it's a very tiny part. It's not photosynthetic anymore. And that seed is going to be an important part of how we think about the gametophyte in these seed plants. In terms of the gymnosperms, the name means naked seed. And what that refers to is that the seeds are produced in female cones like you can see here. This structure is called a scale, and the scale is where that female gametophyte is going to be produced on this plant. Just to give you the phylogenetic perspective on it, here you can see some of those important lineages, the club mosses, so the lycophytes, the ferns, and then we have our gymnosperms here those different lineages. But you see that this is not a monophyletic group, because when we talk about gymnosperms, we're excluding the angiosperms. So seed plants is what we think about if we really want to keep it into a good monophyletic group structure. But realize that the gymnosperms are specifically referring to these plants that produce seeds, but not flowers and no fruits. So here are some examples of the different groups. So the conifers, the pines, that you can see here. Typical ponderosa pine here, good example of these with their female cones. So the woody cones you think of as pine cones, those are female structures. And then these much smaller pollen-producing male cones. Some other examples of conifers, things like Douglas fir that we find a lot of in the Pacific Northwest, junipers, which are a common shrub and tree in some places. This is a cycad, picture of one here. They look like palm trees, but they're not. They do have that big sort of type of leaf, but when we see the cones that they reproduce, with, they're clearly gymnosperms. Ginkgos, these are a very common landscaping tree. It's the only species in this phylum. They have these flattened out leaves like you can see here, and the cones, or some folks describe them as fleshy seeds. That's what's produced here. This is on a female tree, and then there are male trees. These are the better ones to have for landscaping, because these fleshy cones or seeds from the female trees, they have a really bad smell whenever they fall off the tree and start to rot. The gametophytes, that one lineage I told you about, if you've ever been out in the western United States, you might have been seeing ephedra, or it's also called Mormon tea. That is an example of a gametophyte. In Africa, there's this plant Welwitschia, which is a gametophyte, and then in the southern hemisphere, this plant Needham is another example of this group. So there are these really interesting plants in that clade. But getting back to the gymnosperm life cycle, we can see what's happening here is we've got our sporophyte. So this plant is going to produce pollen-bearing cones and ovulate seed-bearing cones, so male and then female. And on the scales of this cone is where the microspore mother cell is going to be. So we now call these spores microspores because they're going to make pollen. And so you can see how it goes through meiosis, eventually going through this process to give us a pollen grain. On the other side, in the ovulate cone, this megaspore, it's going to go through meiosis to eventually give us one surviving, megaspore mother cell will give us one surviving megaspore that will become contained in the female gametophyte. And what happens is we get pollination. So a pollen grain lands here. A pollen tube is going to grow down through the female tissue. So that's what's happening here, as you can see the pollen tubes growing. And there's a tube nucleus. It's controlling the physiology and growth of that pollen tube, and a sperm nucleus. And that sperm nucleus is going to do the fertilization. So this tube grows in here. Whenever the tube reaches the egg-containing structure, the archegonium, it's going to release that sperm to fertilize the egg and make our new gametophyte. What's important about this is that inside of this seed, we've got a sporophyte and then all of this gametophyte tissue, and that's what's going to feed that seedling whenever this seed lands somewhere and germinates. So in the gymnosperms, the female gametophyte, just like in our seedless vascular plants, just like in our bryophytes, that new sporophyte is nourished by the gametophyte tissue. Here's some pictures to show you what that looks like in micrographs. So here, if you've got allergies, this is the scourge that you have to live with. But this is the male cones. Here are the scales that produce these sacs full of pollen. And here's a close-up of the pollen grains. You can see the tube nucleus and the sperm nucleus inside. And then also these two little air bladders. This is really characteristic of pine pollen, which helps carry it on the air. The female gametophyte, things get more complex here. So this is a cross-section through a female cone, so one of these woody cones like you see here. It has these outer layers called integuments, and inside of that, that's where our female gametophyte is. Inside of that female gametophyte tissue is where we've got our developing megaspore that will contain our egg nucleus. And you can see that process of how all of this is developing. The pollen grain lands, and again, fertilization. Pictures down below show how all of that actually looks in real life, but you can see those structures there. Here's another way of just thinking about these. So here's a female cone, pine nuts, pine seeds, if you've ever eaten those. This is the female gametophyte tissue there. Sometimes they have a hard outer layer around them, but these structures inside. And this just shows, again, the process here. You can see pollen grains landing, the seed with all this female gametophyte tissue in red, and there's our new sporophyte. All right, our last group here, the angiosperms, the flowering plants. The key thing here is we've got this flower. It's a highly modified set of leaves for reproduction. We're going to produce a seed with these, and the seeds have a tissue called endosperm. That's not found in any of the other groups. And what's important with this endosperm evolution is endosperm feeds that early embryo and not the gametophyte. So we're completely taking the female gametophyte out of that nutritive role. We do have vascular tissue in these plants. We see improvements even in the vascular tissue over what we've seen before. And then finally, we produce these seeds. They're enclosed inside the ovary tissue. So that's what we mean by the angiosperms. It means closed seed as opposed to the naked seed in the gymnosperms. We have mature ovary tissue that surrounds and covers that seed. I'll remind you to look back at some of your lecture notes and previous things we've talked about with the parts of a flower. We just realized that we've got the perianth composed of the sepals and petals. So these two things here are primarily functioning for attraction, pollinators, and protection of the early bud there. But then we have the stamens, which will be making the male gametes, and then the pistil, which contains the structures where we'll have the female gametes being produced. So be sure to review your flower parts. This slide, I just wanted to put this in to show you how the male parts and the female parts of the flower, so the stamens and the pistils, these are just modified leaves. So all the parts that we see in a flower, we're really looking at that same trend, that same pattern we've seen throughout the class in evolution, existing structures being modified for new functions. And you can really see the idea of what we mean by a carpel with this picture, just thinking about a pea or a bean. What that really is just a modified folded over leaf, and we're producing these ovules. So where our female gametophyte is going to be contained, we're producing those ovules inside of that folded over leaf structure. And you can see how all of that is put together down here. So like we've done before, let's just look at the phylogeny first before we get to some of the details of the life cycle. Here we've got two groups we've talked about before, the monocots and the eudicots. But these are some of the older lineages, what we call the basal angiosperms. These do represent some existing lineages of flowering plants. Their seeds do have two cotyledons, so they are dicots. But they don't come from the same lineage in the same way. They aren't monophyletic like the other eudicots. So this is the arrangement of those groups. Here's some pictures of those basal angiosperms, so magnolias, star anise, water lilies, another plant from the southern hemisphere, ambarella, and then, of course, our monocots, which you probably recognize a lot of these plants, and then eudicots, shown here, some representatives of these. The big change that we see in the angiosperm life cycle or alternation of generations is we get a process called double fertilization. We're going to have two fertilization events. One sperm is going to fertilize the egg to make our next sporophyte, but the other sperm, what it's going to do is fertilize part of the female gametophyte to make endosperm. And so that's kind of what you see being diagrammed in this part of the life cycle, the way that the male gametophyte and the female gametophyte are interacting to have this process of double fertilization that gives us a seed that for some period of time will have this endosperm tissue feeding and nourishing that developing embryo. There are a lot of complexities that go beyond that. We're just going to think about a real simple model of how this works. So this figure shows the two things that are happening in a flower. The male side of things inside of the anthers, that's where we have this microspore mother cell or microsporocyte going through meiosis to make, again, pollen grains. What you want to keep in mind is that these pollen grains, this male gametophyte, when it's mature, is going to have a tube nucleus and two sperm nuclei. Over on the female side of things, again, we've got our flower. Inside of the ovary, we have this ovule. So just like we saw in the gymnosperms, an ovule is this integument layer surrounding the female gametophyte. So we have meiosis producing a megaspore here that becomes the female gametophyte. So this whole structure inside This is the female gametophyte. It's made up of 7 cells with 8 nuclei, and it's surrounded by integuments. So this is diploid tissue around that female gametophyte, and together this all makes up the ovule. Whenever we think about double fertilization, what's going to be important is our egg cell, which is right there, and these 2 polar nuclei. Those are the cells that are going to be involved in making the double fertilization event happen. So here we can see an initial diagram of what's happening. So we've got pollination, just like in the gymnosperms. The pollen lands on the receptive female tissues, and a pollen tube is going to grow down through the style. So that's what's happening, is that pollen grain is germinated up on the stigma. It's growing down through the style. That's what's being shown here. And that pollen tube will eventually come to an ovule. Inside of that ovule, one sperm will fertilize the egg, and the other sperm is going to fertilize the 2 nuclei in this central cell. And that's where the endosperm is going to come from. Of course, that process continues on to make the seed and the next seedling. But let's just see the different parts of what's happening. So here's a close-up of a anther. You can see the microspore mother cells. Here, let me change the color of the. Here we go. So up in here, you can see how we've got those microspore mother cells getting ready to undergo meiosis. That process is carrying out over here, and really nice views of the chromosomes separating down here and finalizing the process of making pollen grain. So that's what's happening in these images. On the female side of things, here's a cross-section through a lily ovary. So a classic monocot. We've got the 3 separate lobes there. And what we've got happening inside of here, these are the ovules. Each one of these things is an ovule, and a close-up of the ovule shows the integuments right there, and then the female gametophyte is going to be contained inside of that region.